Hello, and welcome to my URSA Scholars Week presentation over an analysis of the effects of rotenone on Danio rerio, zebrafish, and the PINK1 enzyme to determine possible Parkinson's-like effects on zebrafish morphology and behavior. Here is my principal investigator, Dr. Melinda Coogan. She works out of the environmental science portion of the Baylor Science Building, and she also is involved in toxicological research involving environmental pesticides, including glyphosate, rotenone, and paraquat. Rotenone is a pesticide that works to eliminate insects and fish attempting to consume plants to protect plant species. Runoff from excessive use of this pesticide affects freshwater ecosystems, as it tends to be impacted by environmental synthetics worldwide. Rotenone has been suggested as a compound that can alter the function of dopaminergic neurons. This is due to its ability to actually alter mitochondrial complex 1 and works as an inhibitor, which leads to the formation of reactive oxidative species, also known as ROSs. Danio rerio are zebrafish and make great biological models because of their ability to share similar genomics to human beings, particularly 70%. They're also useful for environmental toxicology, such as this experiment, pharmacology, DNA damage repair, cancer research, and pathological study. These organisms provide prospective researchers with a model capable of serving as an effective detector for environmental hazards as well as toxic effects that are adept at affecting the human populace. Rotenone, as shown in this image on the top left, has a mechanism that allows for fish to easily absorb the compound as well as for it to be metabolized in their system. In the past, these fish have served useful in testing the adverse effects of chemicals on the neurological and behavioral components of a person or of an organism, such as membrane potential in the mitochondria, oxidative activity of the mitochondrial respiratory chain complexes, or the ETC, electron transport chain, reactive species formation, as well as fish behavior. Parkinson's is a neurodegenerative disorder that causes nerve cell damage, particularly dopaminergic neurons, or nerve cells, decreases in dopamine and motor neuron impairment, which leads to the shaking and stiffness that is seen in Parkinson's patients. PINK1, also known as the P10-induced kinase, is associated with Parkinson's disease and has been studied for its therapeutic advances in the treatment of Parkinson's. PINK1 exists as a serine-threonine kinase that is mitochondrially targeted. The Parkin gene is known as the ubiquitin ligase, meaning that it attaches to ubiquitin molecules to substrate proteins. PINK1 mutations alter ATP production by reducing NADH, or nicotinamide dehydrogenase, or complex 1 efficiency, same thing, leading to the formation of reactive oxidative species, which affects the mitochondrial DNA, leading to mitochondrial DNA mutation. ATP consumption actually occurs when, in turn, the electron transport chain is supposed to form a gradient, which then produces ATP. But because of its altered effects on the mitochondrial complex 1, mitochondrial production turns into mitochondrial consumption. And this can all in turn lead to cellular death, also known as apoptosis. And in the top right, second to the right, you can see the mechanism for the serine-threonine PINK1 kinase. The electron transport chain, located in the inner mitochondrial membrane, there are 4 complexes as well as a 5th that produces ATP. Complex 1 is known as NADH dehydrogenase, where NADH is brought into the cell from glycolysis as well as the TCA cycle and is oxidized into NAD+, that's what I meant to say, which carries 2 electrons, and then they bounce through the chain, forming that proton gradient where oxygen will accept the electrons as its final electron acceptor, forming water. Complex 2 is the succinate dehydrogenase with a coenzyme Q, Complex 3 is a cytochrome C reductase. Complex 4 is cytochrome C oxidase. And complex 5 is ATP synthetase, which generates ATP where the protons produced fall through from the gradient back into the mitochondrion, producing 1 ATP per 4 protons that return into the proton pump. Back into the membrane, of course. Because of the way that each of these complexes actually works, when you inhibit one complex, everything that follows is affected, meaning that ATP synthetase cannot function properly if mitochondrial complex 1 is impaired. Continuing on with the background. Zebrafish embryos fully form their organ systems after 1 day post-fertilization. Because of this, generally, observation begins on the second day, where hatching can usually occur 2 to 3 days after the second day post-fertilization, also known as DPF. This allows for us, as researchers, to observe the fish as they develop their organ systems, as well as their circulatory system, including their heart and other various venous proportions of the zebrafish. The larval stage for a normal zebrafish lasts 45 days post-fertilization, and they reach sexual maturity 3 months post-fertilization. But for the sake of this experiment, we kept them simply for 12 days post-fertilization, which allowed us to still obtain critical data needed for the non-lethal dosing of rotenone. Rotenone dosing at 0, 2, 4, 6, 8, 10 micrograms per liter, or ppb, concentration was done 2 to 10 days post-fertilization and were replenished daily to ensure that concentrations remained consistent. Endpoints for the zebrafish for us to quantify any morphological change included length of the zebrafish, spinal angle of the zebrafish, swim bladder area, and ocular distance, all of which were measured after the zebrafish had emerged from its embryo state. The microscope that we used, as seen in this picture, was set to a magnification value of 2, and we used the open source software known as ImageJ to capture and quantify these morphological changes, particularly at 764 pixels per millimeter. Length was measured from the top of the zebrafish's head to its tail. Spinal angle was measured from the top of its head to the center of its body and at the angle by whichever the tail was extending. Swim bladder area was quantified by checking the zebrafish's torso and simply circling a full area using the ImageJ tool to measure the swim bladder area. And ocular distance measured the distance between the zebrafish's eyes as the zebrafish was standing in a dorsal position where we could simply see its body. This study objectively sought to determine the neurodegenerative effects of rotenone through the observation of early development, morphology and behavior through physical endpoints, as well as the magnetic red ball assessment, which was conducted 11 days post-fertilization and 12 days post-fertilization. This allowed us to determine whether their Parkinson's was affecting their locomotive skills, as well as their behavioral and cognitive ability. We believed and hypothesized that higher concentrations of rotenone would induce statistical significance while generating Parkinson's-like symptoms in Danio rerio. The magnetic ball assessment we believed was important because it provided us an external stimulant to show the fish, and also because, as shown in one of the sources that I acquired, the color red, should be a point of avoidance for zebrafish. However, if the zebrafish acted differently towards the red ball, then we knew that early onset Parkinson's, was occurring within the fish's neurological pathways. On average, zebrafish should tend to observe the color and avoid contact. And if we knew that if fish were avoidant, that meant that normal and unimpaired behavior was occurring. 200 zebrafish embryos were ordered from Carolina Biological, with extras ordered as a contingent. Lux was read. Lux using our Lux meter was read from between the 200 and 400 value. A and B strains of embryos were kept from at 27 degrees to 30 degrees Celsius in buffer water solution using reverse osmosis. 60 milligrams per liter of instant ocean salt was added. 1.2 grams of instant ocean salt to be specific, with 10 milliliters of methylene blue to prevent fungal infection, and 20 liters of deionized water. pH was kept between 7.4 and 7.5. Water quality tests were conducted each day to ensure that the salinity and alkalinity, hardness, dissolved oxygen, and nitrogen waste were all kept constant. Rotenone was prepared using 250 micrograms of rotenone powder and 250 milliliters of DMSO to create 1,000 micrograms per liter concentration of rotenone. MS-222 anesthetizing agent was used to allow the fish to be anesthetized while we took pictures of them in the 96 well plate crate. And 11 dpf to 12 dpf behavioral assay was conducted using a 1.35 centimeter red ball as a visual stimulus for avoidant assessment. Statistical significance was assessed through Microsoft Excel, where F and T tests were conducted based off of equal and unequal variance with an alpha value of 0.05 for the p-value. Here are the results for our length versus concentration, with an average length on the left side in the graph and the t test of statistical variance here on the right. For length, for spinal angle, for swim bladder area where the significance was detected, 2 to 4 parts per billion, or micrograms per liter, and ocular distance. Here was the results of the behavioral assay with R squared values of 0.2525 and 0.0239 on 11 dpf, as well as compared to 0.1207 and 0.0239 on 12 dpf, respectively. Significance occurred at 2 micrograms to 4 micrograms per liter of the swim bladder endpoint. However, higher concentrations from what we hypothesized should have yielded statistical significance and development, but however they did not, which brought into question that further assessment needed to be required. An ELISA test, also known as enzyme-linked immunoabsorbent assay, was going to be used regardless to determine how and when PINK1 kinase was being affected. However, now we see that the negative effects of PINK1 can also affect Parkin gene, which is a ubiquitin ligase, which when altered and mutated, can affect and induce Parkinson's-like effects. Parkin's cell-based ELISA kits allow for the detection of Parkin gene using anti-Parkin antibodies and are detected by HIP conjugated secondary antibodies using calorimetry. And here's on the left is an image showing how the Parkin cell-based ELISA operates step by step. Phosphorylation of ubiquitin by PINK1 Parkin system is critical for cellular function and the prevention of mitophagy, and then prevention of early onset Parkinson's disease. Possible ways that we probably would have been able to determine whether the PINK1 gene was causing or mutations to the PINK1 gene was causing Parkinson's-like symptoms could include knocking out the gene that codes for PINK1 and working with a wild type dosed in the 6 concentrations, as well as the knockout dosed in the 6 concentrations over the 12 dpf, including the behavioral assay in the final 2 days. In conclusion, the findings that rotenone at lower concentrations contain significant result as opposed to higher concentration was very puzzling and required a potential re-examination as well as further testing. However, the evidence is irrefutable that bioaccumulation of pesticides indeed causes unintentional cascades in the environment. The toxicity of these commercialized products must continue to be tested and explored as well as identified to maintain public and environmental safety. Regarding Parkinson's disease, there is evidence that rotenone and its ability to bioaccumulate can lead to early onset Parkinson's-like symptoms. With implementation of a motion tracking camera software at a higher frame rate, it would be possible to truly detect the severity of the motor impairment caused by rotenone non-lethal dosing from the beginning of 2 DPO from we begin observation to the 12 days post-fertilization that we aspire to reach. Thank you so much for watching my presentation, and I hope you enjoy this URSA Scholars Week. Have a wonderful day. And quickly before I go, here are my references.